Good morning, everyone. Welcome to today's lecture with Dr. Didier. He will be your instructor today. Sit tight as we dive into the lecture.
Hello, you're welcome to today's lecture. Today, we shall be looking at histology of the muscle tissue. By way of introduction, the muscle tissue is one of the basic tissues specialized to shorten in length by contraction, causing movement. That is to say, that muscles have cells that have the special ability to contract or relax in order to produce movement of the body parts. It is via contraction of muscles that virtually all movements of the body are ultimately produced. Let's look at general features of muscles. Muscle tissue is made up basically of cells called myocytes. These cells are elongated in one direction and are therefore often referred to as muscle fibers. Each muscle fiber contains contractile proteins called actin and myosin. Each muscle fiber is closely invested by a delicate connective tissue called the endomysium, which supports muscle fibers and unites them to each other. The endomysium is continuous with that around other muscle fibers. Hence, the force generated by different muscles or muscle fibers get added together to generate a greater force. This connective tissue framework also provides pathways along which blood vessels and nerves reach the muscle fiber. A bundle of muscle fiber is called a fascicle, and a fascicle is surrounded by a stronger sheet of connective tissue called the perimysium, while the entire muscle is surrounded by the epimysium. The various organelles of muscle fibers have been given special names. The plasma membrane is called the sarcolemma. The cytoplasm is called the sarcoplasm. The endoplasmic reticulum is called sarcoplasmic reticulum. And the mitochondria is called the sarcosome. There are different types of muscles, and these are based on the following classification. Based on the presence or absence of striations, we have striated muscles. Examples here include the skeletal muscles, which are muscles that attach to bone, and the cardiac muscles present exclusively in the heart. We also have the non-striated muscles. These are smooth muscles, and they include muscles of viscera or organs of the body, and muscles of the vascular system. These include the arteries, the veins, and the lymphatics. We also have classification based on function or histology into skeletal muscles, cardiac muscles, and smooth muscles. Another basis of classification is based on the type of control. Under this classification, we have the voluntary muscles. Most skeletal muscles are under voluntary control. Voluntary in the sense that their actions are under your choice. Your leg bones won't contract and make you to sit up or kick a ball until you choose to move it. We also have the non-voluntary or involuntary muscles. These are smooth muscles, and they are under involuntary control. Note that the contraction of the heart is inherently rhythmic. The rhythmicity is determined by a pacemaker located near the SA node, from where the impulses spread to the entire heart through the conducting system of the heart and cause the various chambers of the heart to contract. But the rate of contraction is modified by the autonomic nervous system. Let's look at the various features of the various muscle types under the light microscope. Skeletal muscles. Under the light microscope, the skeletal muscles Their fibers are long and cylindrical without branching. Each fiber is a syncytium, that is to say that they are multinucleated and formed by the fusion of many individual cells. Their nuclei are elongated and lie along the periphery of the fiber, just under the sarcolemma. The sarcoplasm is filled with numerous longitudinal fibers called myofibrils, and it is these myofibrils that push the nuclei to the periphery. There is the presence of transverse striations, and this is the most striking feature of the skeletal muscle. After staining with hematoxylin stain, the striations are seen as alternate dark and light bands that stretch across the muscle fiber. The dark bands are called A bands, while the light bands are called I bands. The cytoplasm, or the sarcoplasm also contains the usual cell organelles. The mitochondria are elongated and numerous. There is substantial amount of glycogen. These provide the energy needed for muscle contraction. There are many nuclei, and there is an elaborate system of endoplasmic reticulum which are found in between the myofibrils. Each muscle fiber is covered by a plasma membrane called a sarcolemma, which is in turn covered by a basement membrane which connects the muscle fibers to the fibers of the endomysium. The cardiac muscle under the light microscope have some similarities with skeletal muscles, but differences exist. The similarities include that the cardiac muscles are made up of elongated fibers with myofibrils. The fibers show transverse striations because their sarcomeres are organized in a regular pattern of alternating dark and light bands, much like the skeletal muscle. The connective tissue framework and the capillary network around cardiac muscle fibers are similar to those in skeletal muscles. They have actin and myosin filaments, sarcoplasmic reticulum, numerous mitochondria or sarcosomes, and other organelles, just like the skeletal muscles. The differences include that the fibers of cardiac muscles do not run a strict parallel formation, but branch and anastomose with other fibers to form a network. The junction between adjoining cardiac myocytes is seen as dark transverse fibers running across the muscle fiber. These are called intercalated discs. Each cardiac muscle is not a multinucleated syncytium, but a chain of cardiac muscle cells, each having its own nucleus, unlike the skeletal muscle. The nucleus is centrally located. The myofibrils are relatively few and merge with each other. Hence, the striations of cardiac muscles are not as distinct as that of skeletal muscles. Finally, let's look at the histology of the smooth muscles. The smooth muscles are made up of long spindle-shaped cells having a broad central part and tapering ends. The nucleus is oval or elongated and lies centrally. There are no striations. This is because the myofilaments are not arranged regularly along the length of the spindle-shaped cell. The cells are usually aggregated to form bundles called fascicles and are further aggregated to form layers of varying thickness.
To get the comprehensive lecture note, comment "I love this lecture" and you will get one. Thank you for being with us to the end of this wonderful lecture. We wish to see you in the next lecture.